Okay, so hello everyone, my name is Yanis Asloudj, and today I will present you an interactive visualization tool we have developed for multi-resolution single-cell RNA-seq clustering analysis. This work was conducted with Frédéric Lalanne, Fleur Mougin, Patricia Thébault, and Romain Bourqui from the Computer Science Lab of Bordeaux, and also the Bordeaux Population Health, where Fleur Mougin and I also work. So basically, in 2025, we have published a new ensemble algorithm for single-cell RNA-seq clustering analysis. It leverages multiple clustering methods to predict some robust clusters, and its results include multiple resolutions as well as certainty values. And so we wanted to investigate the results of our clustering analysis using some sort of visualization system. Unfortunately, as you can see on the left of our poster, the main approach used today for single-cell data science visualization is scatter plots. So basically, what you often see in single-cell transcriptomics is that you will get some data matrix. You will use some nonlinear dimensionality reduction approach, such as t-SNE, UMAP, ISOMAP. And then you will project the two main dimensions on the scatter plot to visualize your data. Unfortunately, this approach is lacking some key aspects, in our opinion, for single-cell data science. First, using a scatter plot, you can't really indicate, well, which marker genes belong to each cluster, right? You can't indicate the certainty behind each cluster you have predicted. And finally, if you predict clusters across multiple resolution, a single scatter plot is not sufficient to represent this aspect of the data. There are other key issues with scatter plots. So for instance, recently in the literature, there are many authors, there are multiple, I guess, authors that criticize the sensibility to hyperparameters of scatter plots. And there are also many authors reporting occlusion problems when you use scatter plots. So basically, because multiple points can share very similar coordinates, they can overlap each other. And due to that, it can be sometimes misleading when you try to look at cluster sizes. And so really for our objective, scatter plots were not sufficient. In order to address this issue, we have investigated if circus plots could help us visualize the data that our algorithm generates. And so, as you can see on the right of our poster, we have developed a new visualization system according to Schneiderman's mantra. So Schneiderman said that a visualization system should first give an overview, right? And this is exactly what our circus plot is about. So on a circus plot, what you can see is that there are multiple sectors. So a sector corresponds to one of these boxes. And each sector encodes a cluster. The radius of each sector encodes the cluster sizes. So we already addressed one of the limitations raised earlier. But also, for each of these sectors, we encode the certainty of the clusters using some graduated scale that ranges from 0 to 1. We also report inside of each sector the marker genes of a cluster, and we further use some combination of tags to indicate if a marker gene is specific to a cluster, specific to the lineage of this cluster, or actually shared across multiple cluster lineages. Finally, we use the coordinates of the sectors to encode their resolution and also their hierarchical relationships. If we look here at the green clusters that are located basically above this gray cluster, we know that they are subclusters of the gray one because they share similar coordinates. But because they are on the outer layer, we know that they are more informative. We know that they are at a lower resolution, right? Another key aspect of Schneiderman's mantra is what is called zoom and filter. Basically, you want to be able to zoom on your data, on your visualization system, and you want to be able to filter some aspects of your data. Using our visualization system, you can do both. So upon clicking on a cluster, you can apply a fisheye effect to zoom on it, which you can see here on the left. And this fisheye effect can be useful to have a better overview of the market genes of your cluster. You can also use a certainty filter to filter out clusters that are very uncertain. And in this case, you use some sort of threshold, and you reduce the opacity of clusters below the threshold. Finally, a third key aspect of Schneiderman's mantra is details on demand. Here, with our tool, if you click on a specific cluster, maybe the blue cluster one, you will open a specific view next to the overview. On the specific view, you have information regarding the certainty, the size, and also the number of marker genes in your cluster. You have information regarding the clustering methods that were integrated together to predict your cluster. You also have a genetic ranking using log-fold changes of your marker genes, which indicates, which marker genes are heavily expressed in your cluster. By hovering a bar, you can know which cluster on the overview share the same marker genes. By clicking a bar, you open a PubChem entry of this marker gene. And we also have a semantic ranking mode, which uses the same approach. And yeah, it's very useful because you can actually look at different annotation sources that associate your genes to different concepts. And by clicking on one of these bars, you open a web page entry that documents this concept. And by hovering a bar, you can see which other cluster share the same concept on the overview. So yeah, basically we have developed a new visualization system that is suitable for our multi-resolution single-cell RNA-seq clustering analysis. And the next step will be maybe to integrate some semantic information on the overview. These are some kind of discussion we have lately. And so thank you again for listening to this presentation.